This video will cover Chapter 13, Chromosomes Mapping and Meiosis Inheritance Connection. Specifically, we'll look at sex linkage, sex chromosomes, exceptions to the chromosomal theory of inheritance, genetic mapping, and a few selected human genetic disorders. The chromosomal theory of inheritance was kind of introduced to the world in the early 1900s. Walter Sutton was the first scientist to really explore the pairing and splitting that was going on in meiosis, the understanding of having chromosomal material. Thomas Hunt Morgan, however, really built on Walter Sutton's work. He worked in the Flyboys lab. Sounds like a silly name, but that's what they called themselves. They bred thousands of flies, and what they were looking at were mixes of red eyes to white eyes. In the F1 generation of these flies, you saw the typical presentation of dominant to recessive. In the F2 generations, however, what they started noticing was that only the males had the red eyes or that recessive trait. They weren't really sure at the time what was going on. Now we better understand it, and we will come back around to see exactly what Thomas Hunt Morgan had discovered in his lab. Sex chromosomes have a lot to do with what he was seeing. Over on the left, you see human sex chromosomes, a rather large X and a teeny tiny little Y. The Y is actually that small. Because there's only one Y, if there's anything wrong with it, the male body has started cutting out those sections of genes because you absolutely need certain parts of your Y chromosome to function. So if any part of it is mutated, they get rid of the bad part, kind of in a last-ditched attempt to save the best part. Not every organism that you come across in biology has this XY setup. Over on the right, you can see a chicken compared to a rooster. They actually have something called a ZW and a ZZ conformation. Bees are in the middle. A female bee has 30 chromosomes and 2 X's, while a male bee might have 30 chromosomes and XY, or they might just have 15 chromosomes and a single X. So their sex determination can be quite different. And then you see the grasshoppers on the bottom. It's kind of similar to the bees where they only have one sex chromosome. A female grasshopper is XX and a male grasshopper is just X. So there are lots of different situations where you see sex chromosomes in variable numbers. Usually it is the female that either has more sex chromosomes or more set rules for their sex chromosomes, and we tend to tie this to the reasoning that the female body has to be able to carry and develop a fetus, so they have a slightly more complex genetic code. We study some diseases that are known to only follow the X chromosomes in individuals, and what this quite a large pedigree is showing you is all of the hemophilia that existed in the royal family, and you actually end up seeing, if you look, we're actually going to start at the bottom so you can see what's going on. If you look at the bottom, the British royal house, the Persian royal house, the Russian royal house, and the Spanish royal house all came from one large initial family that you see up top. And there's one individual, and her name is Queen Victoria. And a lot of, well, essentially all of the hemophilia in these lines came from Queen Victoria. So you can see that in her space, she is a circle that is half filled. We've looked at pedigrees before, but not quite what it means to be half filled in. So anytime you have that situation, what it is attempting to show you is that that individual with the half-filled circle, I'm not sure why it's deleting what I write, I'm sorry, with the half-filled circle, they are simply a carrier. So they have the ability to pass on the genes, but they themselves are not carrying, are not ill, or showing that particular phenotype. So Queen Victoria was the original carrier, and you notice that she gives it to a daughter, Alice, her son Leopold, and her daughter Beatrice, and it just kind of explodes as the family goes on, because there's lots of essentially interbreeding in royal families as they tried to ensure that their bloodlines remained pure. So you see an affected child here, 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 And you can kind of trace it on down the line, and we can even figure out who our carriers are as we go. Some of these families are quite famous for being sick. One of the most famous families for being sick is this little line right here in Russia, because the fact that Tsar Nikolai, his son, had hemophilia, they think that that's one of the reasons that the revolution happened. The people felt unstable in their potential leadership, and it kind of caused a regime change in Russia. So pedigrees can be very interesting historically to study, and you can see how genetics makes a big difference, even in the history of a country. We do look at different genes and their effects on the body, and some people say, well, if you have an extra chromosome, if you have an extra chromosome 21, we know that that is Down syndrome. If you have extra sex chromosomes, that can be very dangerous. Well, why aren't women sick? Because all women have 2 X chromosomes. Well, this little circle right here inside this white blood cell is called a Barr body. And this Barr body is actually an X chromosome that has collapsed on itself and is no longer capable of performing any functions. One X chromosome in each somatic cell of a woman's body will essentially shut itself off. And it's a dosage compensation. You only need one functioning X. Having 2 can make you ill, so we turn one off. One of the easiest ways to look at that in a biological sense is to look at a cat. Really basic picture. So you see over on the left in the early embryo, the X chromosome carries the alleles for the fur. One allele in this cat is orange, one allele in this cat is black. Those early cells in the embryo are going to divide and divide and divide and to eventually become our whole adult cat. Well, in some of the cats, one of those X chromosomes that's getting turned into a Barr body might have contained that black allele, and it's going to be destroyed. So those cells are going to exhibit the orange allele. On the other hand, some cells could randomly choose to inactivate the X with the orange allele, so those cells exhibit the black allele for fur. And that's how you end up with these tortoiseshell cats. They're always female, and now you know why. It's because of dosage compensation in sex chromosomes to keep those individuals healthy. We focus on some of these ideas of not so much sex chromosomes, but passing on diseases through maternal inheritance. And we can pass on any diseases related to our mitochondria through our mothers. And that's because, of course, our mitochondria only come into our bodies, into our embryos, from the ones that were helping keep the egg cell alive. So this Leber's hereditary optic neuropathy is a disease that has to do with eye health. It's of course maternal in its inheritance, and it's actually the NADH dehydrogenase in the electron transport chain in individuals with this disease is it's inhibited. It doesn't function terribly well, so they become quite sick, and their optic nerve can actually die. So both males and females are affected by this disease because everybody has mitochondria, and every healthy person has the same number of mitochondria, but because it's passed on maternally, only women who are affected can give it to their children. Men are not capable because they have no mitochondria in their sperm. All the mitochondria for their potential children come from their wife. We can map all of these alleles onto chromosomes. We've gotten a little bit better at it. The confusion comes from the fact that some genes are on the same chromosomes, but they don't always necessarily travel together. We've already hinted at this in class, and it's because of crossing over. 2 homologous sister chromosomes can exchange either arms or legs, and you switch where the genes are located. So you always have a gene for a particular particular trait about you, but you might have a different allele. And these changes in alleles and where these alleles lie can be traced via genetic mapping. Stop and think for a second before we get into our linkage maps. Do you think that genes that are located closer together would be separated more or less often by crossing over? Hopefully your answer to the question was less often. They're separated less often, because if you think about it, if this is your whole chromosome, if you have a gene right here and you have a gene right here, there is a lot of potential space where those two things could be cut off. If you have a gene here and a gene here, though, the odds of choosing a number in this teeny tiny little space is quite small. And that concept leads us to study things like linkage and data maps. So linked genes are genes that are very close together, so they often travel together. The recombination frequency in genes is a number that we can calculate. Sorry, that was bugging me. It's a number that we can calculate, and it tells us how often genes seem to be traveling together. So we look at the recombinant progeny over the total progeny. What I mean by that is, if we were curious how often gene A and gene B traveled together, we would breed a whole bunch of potential progeny. We would count all the progeny that had gene A and gene B, and we would divide that by the total number of progeny that we had in our entire system. And that's going to tell you their recombination frequency. When we talk about creating linkage maps using linkage data, we actually just convert our percentages over to map units. That should say map units, I'm so sorry. We convert our data over to map units. One map unit is called a centimorgan, and it's after one of the scientists that worked in that Flyboys lab, because he's the one who realized this process. And there's a lot that we can do with these linkage maps. So to construct a linkage map, you have to cross many heterozygous individuals, and then you count the progeny. Once you get them counted up, you divide the individual genotypes by the total number of individuals in your system, and that will give you the percentage of crossing over events that happened in each of those meiotic divisions. You have to remember, though, that double crossing over can occur because different chromosomes are coming together quite a few times in each division. So the genetic differences in linkage maps are probably largely underestimated. Sometimes we do use 3-point crosses, so we're looking at 3 different genes, and it allows us to watch the alleles a little bit more closely so we can really see when they're recombining differently. So here's what that concept looks like. So up top you see meiosis. If there was no crossing over between genes, then you're going to see what the chromosomes look like on the left and then the products look like on the right. On the bottom, it's showing you an example of crossing over. So on the left-hand side, you can see that the original chromosomes were big A, big B, and the bottom chromosomes were little a, little b. With one crossing over event, you change one chromosome to have both a large and a small letter on it. So this is your basic chromosome mapping. When you add in a third gene, you essentially are capable of capturing more changes and ensuring that if there is a double crossover event, you're more likely to notice. So up top we have ABC in capitals, ABC in lowercase, and then you see in the middle you're switching out the large B for the small b, and A and C are staying in the same place. if you were only looking at A and B, if there was a change to C, you might have missed that. So by examining even more chromosomes and examining them in a longer line, you're capable of catching errors. Because if we were to expand this to say A, B, C, D, and E, you might have a double crossing over event where A and B both changed, but then in the next crossing over event, B changed back. But because all of these potential variabilities, it sounds like a lot, all of these potential variabilities probably would have changed genes down the line, you're going to notice your changes. So maps in the human genome, really, lately they've all come from historical data. We've been looking at genes that we can trace. We've just been able to kind of cheaply map genes, so we're just starting to do this in families. There are some markers that we're looking at. The markers are always tied to different phenotypes, but it can be quite hard to find exactly where those markers are. So we're not entirely sure where the marker for fingernail shape lies on the human genetic code because it's so huge, and a lot of our genes exhibit pleiotropy and polygenetic inheritance. So they've kind of picked anonymous markers as well. So long, either really, really long stretches that make people unique individuals, or they've started picking out single nucleotide polymorphisms. We call them SNPs. And they are just very specific changes in individuals. You are 10 million SNPs different from the next human being that you will see, even if you are related to them. Sometimes we have very long similar codes, and for whatever reason, your body has replaced one of those nucleotides, and we're capable of tracking them. So as we're making maps in the human genome, it's not as simple as tracking A, B, C, D, and E to look and see when things are switching, maybe once or twice, going from their original position to a new position and back to their original position. This is what some of our linkage maps are looking like in various human chromosomes. I realize this looks kind of crazy. That was my intention, to show you just how busy these chromosome maps have become. We're getting really effective at picking out genes that carry out specific tasks for us. A lot of the ones that we're most familiar with do cause a variety of illnesses. We have come up with a lot of human genetic disorders. Some common ones that you hear about quite a bit: cystic fibrosis, sickle cell anemia, Tay-Sachs disease, hemophilia, Huntington's disease. Some of these are very, very common. And we will take a closer look at them in class via a homework assignment, or your homework assignment might be online for you, depending on which section you are in. As we look at all of our chromosomes, we have to kind of remember that they are all situated quite differently. So here are some of the potential shapes for chromosomes. The telocentric, all the way to the left, those are some of your smaller chromosomes in your body. So those would be the ones in the late teens and 20s. The acrocentric chromosomes would be like the early teens. Submetacentric chromosomes would be maybe chromosome, I would say probably 8 through 13, I think, are submetacentric. And then the metacentric chromosomes, those are your really, really big guys. Remember the way that your chromosomes are put in order? We say that we have 22 pairs of somatic chromosomes. 1 is the biggest, 22 is the smallest, and then your 23rd pair are just those sex chromosomes. So I put this up here. We're going to talk about some genetic disorders. And some of the references discuss the P arm and the Q arm. So the smaller arm of the chromosome, or the arm that we consider the top, that is called the P arm. The arm that's either longer or on the bottom is considered the Q arm, because in genetics we need to be sure to mind our P's and Q's. I'm not kidding, that's where the name came from. So as we look at some of these genetic illnesses in class and you hear things like 15q deletions or 15q repetitions, we're talking about chromosome 15. and the long arm is what either is missing something or has something a bit strange to it. Non-disjunction in chromosomes is where a lot of our genetic illnesses come from that we are most familiar with. So in your autosomes, chromosomes 1 through 22, you can have either a monosomy or a trisomy. Monosomies is when you only have one of those given chromosomes. So you only have one chromosome 3 or you only have one chromosome 7. Those extraordinarily rarely make it past embryonic development, and even if they do, they miscarry the vast majority of the time. We're talking like 99.9 repeating percentage of the time. Embryos that have monosomies will not become even viable fetuses. Trisomies, on the other hand, are quite different. The trisomies that are known to exist in living individuals is trisomies 13, 15, 18, 21, and 22. The larger trisomies don't usually survive to adulthood. When I say the larger trisomies, you have to remember that trisomy 13 and 15 are larger trisomies than 21 and 22 because 13 and 15 are the larger chromosomes. Smaller trisomies, however, are surviving longer and longer. Trisomy 21 is the one that we hear about most often. That's Down syndrome. We're doing a better job at addressing the potential healthcare issues in these individuals when they're very, very young, and it's giving them better outcomes on life. Non-disjunction of sex chromosomes gives you a very interesting set of potential illnesses. So up top you have Klinefelter syndrome. So this is an X and a Y. People with Klinefelter syndrome, they're males, but they have secondary sex characteristics. XO is Turner syndrome, so you only have one X. So these are females, but they usually don't undergo puberty and they're missing female sex organs, although they appear to be female, or they are female. They're female externally. Individuals that are XYY have supernumerary Y syndrome, and this one is really interesting to me because some of these individuals tend to show a lack of control over impulsive behaviors. And what they have found is that XYY syndrome is really common in the general population, but it's even more common in the prison system with violent criminals. So they think that the extra Y is releasing added testosterone into these individuals, so they have a harder time with impulse control. That of course doesn't make the things that they've done to get themselves in prison any less repulsive, but they're kind of curious if that's one of the things that they should start focusing on. Triple X syndrome is when you have an extra X chromosome. Those individuals are occasionally infertile and sometimes have learning disabilities, but they're usually okay. And then individuals that have XXXY, certain parts to them, they can actually be hermaphroditic. So that's when you have two different sets of sex organs in your body. All of these different diseases all of these different chromosomal abnormalities, I shouldn't say diseases, cause different issues in different people, and you see varying levels of physical and mental disorders. Some really interesting disorders seem to come from genomic imprinting. So where you get the bad mutation is going to make a difference of what symptoms you show. So a phenotype is caused by a specific allele, but the phenotype changes depending on the parent that it comes from. So it's imprinted in one parent, and what that means is that that gene is silent for whatever reason. It's not mutated, it's just silent. It might be methylated, for instance. And then in the other parent, it's damaged. That matters in something like Prader-Willi syndrome. So in Prader-Willi syndrome, you have a 15q11 to Q13 mutation. So chromosome 15 on the Q arm in spaces 11 to 13, you have a mutation. If you get that mutation from dad and your gene is inactive in your mother, what you will find is that they have Prader-Willi syndrome, PW syndrome, and they have really strong obsessive compulsive disorders, extreme food obsession, and elastic skin issues. The big issue with Prader-Willi syndrome, outside of the mental disabilities that these patients experience, is that they have insatiable hunger. So that awful feeling you get in your stomach when it's been about 2 hours since you should have eaten and you start to feel physically ill, PW patients feel that way all of the time regardless of what they've eaten. So a lot of these families have to resort to padlocking kitchens and cupboards and hiding anything edible because these poor patients who don't understand what's going on with them will they'll eat soap if they think that it's going to make their stomachs feel better. So they grow and they can become easily morbidly obese because they also see different issues with their ability to burn fat, to metabolize the fat that they take in. So Angelman syndrome, the second one on here, and I'm sorry that has the red underline, it's going to drive me nuts. Angelman syndrome is the exact same mutation, but in this instance, you get the mutation from mom. And that particular section is inactive in dad. So patients with Angelman syndrome, they don't have the obsessive compulsive disorder so much as they have a lot of brain symptoms, seizure, ataxia, so really jerky movements. They have a harder time at life physically. So Prader-Willi has more mental impacts. Angelman's disease, Angelman syndrome has more physical impacts. It's very, very dangerous to have Angelman. Prader-Willi can be treated with a lot of behavioral interventions. So it's quite interesting to think that a disease can vary so much just depending on where you got the gene from. A lot of genetic testing is done now to help prepare parents and families prior to the arrival of a genetically ill child. Genetic counseling is all about preparation and knowledge. Nowadays, they've been looking at pedigree analysis. So you see more information with understanding that you have a history of Tay-Sachs in your family, a history of muscular dystrophy, a history of sickle cell anemia. They can perform amniocentesis, or they can perform chorionic villi sampling. All of those bottom two consist of taking samples from mom and testing different tissues that are close to the baby. Some big leaps now are fetal blood cell testing, and they actually retrieve the fetal blood cells from the blood of the mother. So genetic testing in early pregnancy has really, really, really changed and is evolving quite quickly as there are advances in medicine. This is the end of chapter 13. We will address any potential assignments and questions in class.